Okay, I think we can get started now. Hello, good afternoon, good evening, and good morning, depending on where you may be in the world. I am Rob Burgess, and I head up business development for Sino Biological, and it is my honor and pleasure to host the next installment in our series hosted by and sponsored by Sino Biological called BioTalk Tuesdays, where we hear about cutting-edge technologies as they relate to biology and therapeutics. We have an excellent speaker today. I think you're all going to really enjoy the talk. We also have a huge number of registrants today, so I'm looking forward to having a great turnout. Our guest speaker today is focused on B-cell response as it pertains to nanoparticle adjuvants. And before we get into the talk today, I just have one housekeeping issue. And that is that I ask everyone, if you have a good question for our speaker, please type your question into the chat box, and we will withhold all questions until the end of the lecture. And then I will run through those verbally with our speaker and get to as many as I can. And while you're at it, please say hello to us in the chat box and also tell us where you're from. We love to hear about all the folks from all over the world, and specifically where people are calling in from. So with that, it is my pleasure to introduce our speaker to you all today. Our speaker is Dr. Wei Luo. Dr. Luo is an assistant professor in the Department of Microbiology and Immunology at Indiana University School of Medicine. His research focuses on understanding the mechanisms regulating germinal center reaction and developing vaccines for infectious diseases. In addition, Dr. Luo investigates the pathogenesis of lymphoma and autoimmune diseases to aid in the development of novel treatments. Dr. Luo obtained his doctorate in biochemistry from the State University of New York in Buffalo, where he studied the ETS transcription factor and its role in B-cell mediated autoimmunity. And the title of Dr. Luo's talk today is Broadening B-cell Response Using a Nanoparticle Adjuvant. Dr. Luo, it's a pleasure to have you today, and we will now turn over the screen sharing to you.
Yeah, Thank you so much, Rob, for the very nice introduction. I really appreciate this opportunity from Sino Biologic to be able to present to people all around the world about my work on vaccine. So I'm from Indiana University Department of Microbiology and Immunology. And the focus of my research is germinal center response that can lead to long-lived humoral memory generation, and how to optimize the memory development to develop next generation vaccines that can neutralize multiple strains of virus. So the title of the talk is Broadening B-cell Response Using a Nanoparticle Adjuvant. So first, I want to briefly introduce how B-cell respond to pathogens or vaccines. So when B-cell encounter a vaccine or the viruses can recognize the antigen or epitope on those immunogens, they can either directly differentiate into short-lived plasma cells or rely on a T-cell dependent mechanism. They can also differentiate into short-lived plasma cells and memory B cells. This is called extrafollicular response. Extrafollicular response can generate a wave of antibodies that has low affinity against the antigen. It's very quick, so you can generate this extrafollicular response within a week encountering the antigen. It's fast, but it's temporary because the plasma cells generated from this response they are all short-lived, and you might get some auto reactive antibodies through actual follicular response, but usually does no harm because this response is mostly short-lived. Another pathway to generate antibodies is through germinal center response. So with T cell help, some B cells can differentiate into germinal center B cells. And in germinal center, there is dark zone and light zone. And the B cells can shuttle from dark zone and light zone to get help from antigen and T cells. And then they recycle to dark zone and proliferate, and they get reselected. And after rounds of selection, they can differentiate into memory B cells or long-lived plasma cells. And this is called germinal center response. And with germinal center response, the B cells can generate very high affinity antibodies, and it also class-switched antibodies, for example, IgG and IgA, and they have specific effector functions. And germinal center-derived plasma cells and memory B cells, they are very long-lived. Some of them can live for decades, so they can provide very durable protection. And germinal center is highly select against self-antigen, so it's more stringent, so you don't really get autoreactive antibodies that easy from germinal center response. This is mostly because the memory generated from germinal center is long-lived, so it has mechanisms to avoid autoreactivity. Because of these features, so it's very important to understand how germinal center works in order to help vaccine design. So a prominent feature of germinal center is affinity-based selection. And you can see that shortly after germinal center forms at this point, and affinity of antibody will sharply increase. Sometimes throughout germinal center response, the antibody affinity can increase 10 to over 1,000 folds. So it's very efficient for antibody affinity maturation. So how does a germinal center do this? So germinal center B cells express a protein called AID, and it can induce somatic hypermutation to the B cell receptor, which is antibody attached on the B cell surface. And because of the mutation, the B cells can either generate a antibody that's of higher affinity, or the affinity might be lower because of mutation. Sometimes it might generate antibodies that can bind to self-antigen. And these different B cells will compete with each other. And the result is only the higher affinity germinal center B cells will survive, and they express a protein called CMYC. And then they can proliferate, survive. And the lower affinity and the autoreactive B cells, they will quickly die. Then you can see after many rounds of selections, affinity can quickly increase. Here is a more detailed model of signaling-based germinal center selection. So when germinal center, the dark zone germinal center cells can bind to antigen, compete with other germinal center cells, they can more efficiently bind to antigen, and it generate a B cell receptor signal. And this signal can help germinal center B cell travel from dark zone to the light zone. And in the light zone, germinal center B cells can interact with antigen presented by follicular dendritic cells, which give germinal center a further B cell receptor signal that can keep them in the light zone. And then they have a chance to interact with T cells, and they present antigen to T cells, and in reward, they get a CD40 or L21 signals. That's the help from the T cells. And with multiple signals, such as B cell receptor signal and CD40, and they can synergistically induce a protein called CMYC, and this can charge the germinal center B cells, and then they can go to dark zone to proliferate and mutate their B cell receptor. And when they proliferate, they will slowly use up their battery. And then they will have to compete because of mutation. They will change their B cell receptor, and they will have to compete with each other. And only the higher affinity B cells can survive this competition, and the lower affinity or other B cells will start to die. And eventually, when there is a solid L21 receptor signal, it usually happens to high affinity germinal center B cells, and then the cells will differentiate into humoral memory compartments, including memory B cells and long-lived plasma cells. And long-lived plasma cells mostly will migrate into the bone marrow and the secrete antibodies for many, many years. So germinal center is very good at affinity maturation, as you can tell, but what they're not very good at is the breadth. So we all went through the past the COVID-19 pandemic. So at the beginning, we get 2 doses of the vaccine, which have very good protection, but over the time, the virus will mutate and generate multiple variants, and eventually the vaccine really doesn't work that well. One of the reasons is the antibodies generated by the vaccine that can bind to the original virus very well, it cannot bind to some of the mutants, such as Omicron variant. So you might ask whether we can generate a vaccine or some strategies that can induce antibodies, such as this antibodies like gold in gold color, that can bind to or most of the variants or all the viral variants. Then it will provide a more broad protection even against future viral variants. I think it's possible because if we can target the more conserved epitopes, even though the virus can mutate, but there is variable epitopes and there is conserved epitopes. If we can target the conserved epitopes, then this will become possible. However, in germinal center, it's not very good at doing this. The reason is because of immunodominance. So what is immunodominance? I'm hoping to use this slide to explain it a little bit. So imagine there is a complex antigen. There is just two epitopes, epitopes A and epitopes B. Epitope B is subdominant, epitope A is dominant. If I get a immunization with just epitope B protein, I can actually generate a robust germinal center response, and which will produce a lot of effector cells, generate a lot of antibodies targeting epitope B. However, if I'm immunized with epitope B attached to dominant epitope A, that is better at inducing B cell activation and activating germinal center B cells. What happens is the dominant response targeting epitope A, which is a dominant epitope, will suppress the subdominant epitope B response. As I showed you in the last slides in germinal center, they really have to compete, and for some reason, the epitope B targeting germinal center B cells, it's very hard for them to actually compete with the dominant germinal center B cells, and they either will die and they can't survive in germinal center, or even they can survive germinal center, and it's very hard for them to actually differentiate into plasma cell, antibody secreting plasma cells. And then eventually I end up making most of the antibodies targeting the dominant epitope, and with very few antibodies that can target the subdominant epitope. This is not because I cannot generate because antibody targeting epitope B. In fact, I can if I just get immunized with epitope B. That's because for complex antigen, some epitope can actually suppress the response to the other epitopes. This is called immunodominance. And this is exactly happening to our response to influenza vaccines. So we get the flu shot, which is an inactivated influenza virus. So it has more conserved epitope. For example, it's a neuraminidase, NA. But because hemagglutinin HA is highly immunodominant, so it actually suppresses the response to NA. So the antibody generated in our body mostly target the HA, but not the more conserved NA. And HA, we know, is highly variable and mutable. And within HA, there is actually conserved epitopes, subepitopes within HA. So HA has this head domain and also has this stem domain. The head domain, if you look at the head domain, it's very variable. So the white are the regions are the most variable epitopes, and light blues are less conserved, and the dark blue are most conserved. So the most conserved epitopes are actually most located in the stem region, and the head region is highly variable. However, the antibodies actually targeting HA are mostly targeting the head region of HA, but not the conserved stem region. So there are some studies measuring the antibody levels in humans. So in children, they have limited exposure to influenza virus actually. So in adults, this is probably higher, even higher. So in children, the serum from children, they have around 100 to 500 microgram per mil of antibodies targeting HA. That's the whole protein. But within this, only 0.1 microgram per mil antibodies actually targeted the stem region. Even though every time we get flu shot, this might increase a little bit, but because the basal level is so low, it's really hardly can do anything protective. So with this, we know that the head region is highly variable, and it's really created a problem because all the antibodies, most of the antibodies are targeting the head, and the virus can actually mutate their head region and just escape the antibody-mediated protection, and the vaccines can no longer work. In fact, the reason why this part is highly variable is probably because it's the virus evolve with the immune system. They try to escape the antibody targeting. So ideally, if we can generate an antibody response, not only target the dominant head region, but also target the more conserved stem region. And in this case, even though the virus try to mutate some part of the HA, but we still have a pretty good protection. And beyond this, there's some other problems associated with immunodominance. For example, the protection mediated by antibodies mostly from neutralization, because the virus actually use their surface proteins such as a spike protein in SARS-CoV-2 or envelope protein in HIV virus. They attach to the receptor on the cell surface and get internalized and infect the cells. If antibodies can block this process, either attach to the surface or the uptake of the virus into the cell, it can neutralize the virus. This is called neutralizing antibodies. But not all the antibodies we generate are neutralizing, because some of the antibodies we generate, they can bind to the antigen, but they don't really block the uptake of the virus and block the infection. And when non-neutralizing antibodies is highly dominant, such as in the HIV vaccines, we currently don't have a working HIV vaccine. It is because HIV envelope protein can generate robust antibody response, but most of those antibodies are not neutralizing antibodies. And it's very hard to achieve a high titer of neutralizing antibodies, let alone if we want to make a working HIV vaccine, we have to generate broadly neutralizing antibodies, even more difficult. So that's why we still don't have a HIV vaccine. And in some other cases, can be even worse. It can mediate antibody-dependent enhancement. It can make things worse because if the antibodies can bind to the virus and it's not neutralizing, first the virus can still infect the cells, and also the antibody can actually bind to Fc receptor expressed by many of the myeloid cells, and the virus can use this to infect myeloid cells. The myeloid cells can travel around the body to spread the virus, and actually this is not good. So to overcome this, we also have to deal with immunodominance. So how to deal with immunodominance to increase the breadth of B cell response? There are a few strategies people have been trying. So the first, apparently, how about just we use the subdominant epitope as immunogen to vaccinate people, right? For example, we use NA from influenza, or we just use the HA stem region, or we call headless HA. We just use those as antigen, immunogen to vaccinate people. Actually, there are some clinical trials using these antigens to make a universal flu vaccine. But the truth is, this approach can work. I think it can work, but in long run, it's probably not going to resolve the problem because it doesn't change the nature of immunodominance. Because if you just immunize with NA, within NA, there are still immunodominant epitopes and subdominant epitopes. And when time goes on, and the virus might actually mutate. If there is immunodominance, they can actually mutate even this subdominant epitope to avoid neutralization. So the second strategy is just design a antigen or immunization strategy, or use chimeric antigen that share the subdominant epitope, but with varied dominant epitope and with prime and multiple boost, and try to focus antibody response against the more conserved but subdominant epitope. I think it will work. And also the mosaic antigen that's attached all these different antigens on one particle, so to kind of cross-link B cells that can target the more conserved epitopes. I think these strategies are very good and they can work, but it also involves sophisticated antigen design, and it may not be good for pandemic preparation. For example, SARS-CoV-2, and when we have this pandemic, it's very hard to predict the emergence of those variants. Without those variants, it's a little hard to make this antigens, and it's for protein engineering. It's also not as quickly can deliver to people to fight pandemic. So the third approach probably is more simple or straightforward. Is just use some adjuvant to overcome immunodominance that can generate antibodies against the subdominant epitopes. However, people have developed many different adjuvants. They either do not work as well, which is they kind of overcome immunodominance, but it's not to a degree that can help protect people, or it's too strong and it's just they can generate toxicity, and if it's not tolerable, and it's not going to get into clinical trials. So an ideal adjuvant, I think, can overcome immunodominance. It can specifically target the lymph node to activate the cells locally, right? And hopefully this adjuvant can increase the germinal center recruitment of those cells, B cells, targeting the subdominant epitopes. So they have more subdominant B cells to start with within germinal center. And in doing this, it probably can have to provide additional help signals to help those B cells, because they are excluded in germinal center, if you want to include them, you probably need extra help. They probably need extra help. For example, toll-like receptor signal, which can activate B cells, might be able to resolve the problem. And within germinal center, and this adjuvant hopefully can promote the selection and the effector differentiation of the subdominant the germinal center B cells targeting the subdominant epitopes. A good strategy is to, if you can boost TFH, which is the helper T cells that can generate signals to help B cell selection and differentiation, or if you can restrain follicular regulatory T cells, those are regulated T cells that can restrain the B cells, can inhibit the B cell response. So, and with all this, and you have to keep in mind that this adjuvant cannot generate a systemic inflammation or autoimmunity or some severe side effects. Otherwise, nobody wants to get those adjuvants. So to develop this kind of adjuvant, I collaborated with a bioengineer, Dr. Qian Yin, and she has this nanoparticle that's kind of unique. So you can, it's self-assemble nanoparticle, and you can embed a TR7 ligand Gardaquil mode into this nanoparticle. And the size of the nanoparticle is a 70 nanometer, and it's ideal for a lymph node retention. So this is a nanoparticle. So I'm not a chemist. I don't fully understand how this works. So I draw this simplified nanoparticle. Basically, it's a slow release TR7 agonist, and it's also controlled release because it doesn't. If the pH is 7, right, it's red. It doesn't release very quickly. But when the pH is lower, it can release more quickly. So this is very good because for antigen presenting cells, when the antigen presenting cells uptakes this into endosome, when the pH drops, it might actually release this within the antigen presenting cells to activate the cells a little better. So first, with this nanoparticle, I'm just curious how the nanoparticle will traffic, or how about the retention of this TR7 nanoparticle? So we labeled the TR7 agonist with flower core Alexa 647. So with this imaging machine, we can track their trafficking and the retention. And first thing we noticed is that this nanoparticle can retain in the injection site for much longer time. For example, we compared this nanoparticle formulation with alum formulated TR7. It's the same amount of TR7 just formulated in alum, and alum is a traditional adjuvant formulate, and itself can be used as adjuvant because it can stimulate immune cells. And people thought adjuvant can also deposit whatever is observed and it's kind of release it more slowly, but compared alum nanoparticle doing a much better job in retaining the TR7 agonist in the mouse. This is day 1, day 3, and by day 3, you can see the TR7 almost gone in the TR7 alum vaccinated mice, but it's still in the TR7 nanoparticle vaccinated mice. So it's much better retention at the injection site. So it can probably persistently activate antigen presenting cells. And also it specifically targets the lymph node. And if you can see here, for alum, even day 1, you don't see any TR7 agonist in the draining lymph node, and draining lymph node is quite small. And for TR7 nanoparticle, and you can see, but day 1, day 3, and you can see this TR7 agonist signal suggesting their lymph node targeting property. So when they talk lymph node, I'm just curious where is actually located in the lymph node. So we did a histology analysis, and we found that this is a white. Its color is we track this nanoparticle TR7 label nanoparticle, and we found that the nanoparticle actually they mostly target B cell follicles, and especially around the follicular dendritic network, where germinal center forms. So it actually can stimulate locally the immune cells, and there are some nanoparticle also in the T cell zone, but mostly in the B cell follicles. And in line with this data, and we also found actually the B cells can uptake nanoparticle, and this is from day 1 to day 3, and it's very robust uptake of nanoparticle, and also other antigen-presenting cells such as dendritic cells, macrophage, and the monocytes. They can also very efficiently uptake nanoparticle, and I also have data to show that they are also better activated by this nanoparticle. So this is the B cells. We use CD86 as a B cell activation marker, and you can see nanoparticle compared to alum formulation can much better activate B cells. And interestingly, when we label the antigen, protein antigen with Alexa 488 to track where they go, right? If they go with nanoparticle or they don't. So this antigen we didn't embed into nanoparticle, but surprisingly we found that for the cells, for B cells, that uptake OVA, supposedly the mostly OVA specific antigen specific B cells, they also uptook this nanoparticle, and you can see this ratio. For this doesn't seem to be a feature of alum formulation, but it's specific for nanoparticle formulation. This suggests antigen-specific B cells might be activated much better by this nanoparticle. So this leads to the next question. So whether for the B cells that targeting the subdominant epitope, so we know that first it's probably hard for them to get involved in germinal center. So whether with actual TR7 signal, we can push these B cells, subdominant B cells, into germinal center, so they actually have a chance to participate in the selection. And we found first, nanoparticle formulated adjuvant can induce much more robust germinal center responses. B cell 6 marks all the germinal center cells, and this is different scale. But you can see this much larger and more germinal center response induced by nanoparticle. This is 1 week, only 1 week after the immunization. And this is the kinetics. You can see, and it's more robust and more sustained germinal center response compared to traditional alum formulated TR7 agonist. And with a B cell receptor sequencing, and we also show that it actually can increase clonal diversity in germinal center B cells, suggesting some of the subdominant B cells might be able to participate in germinal center response. So how about T cells, which can provide critical help or regulatory signals to germinal center B cells? First, we found the nanoparticle adjuvant can leads to a more robust follicular T cell response. And within the follicular T cells, there are a substantial percentage of those cells are actually expressed FOXP3. So those are not helping cells; those are regulatory germinal center T follicular help regulatory cells. And you can see in alum, it's almost 30% at day 7 or 40% at day 14 of those follicular T cells are expressed FOXP3. But the nanoparticle adjuvant can drastically reduce the ratio of regulatory cells versus helper cells, or increase ratio of helping cells versus regulatory cells, suggesting more help available to germinal center B cells, might help them to survive. So far, we know that it might actually recruit more subdominant B cells into germinal center response, and with the change the robust follicular T cell response and change the ratio of TFH to TFR, there's more T cell help to germinal center B cells with nanoparticle adjuvant. And also, the nanoparticle can also induce a very robust early plasma blast response. And assuming this is response is dominated by its antibody against the dominant region of the antigen, and it might actually mask the dominant epitopes and making the subdominant epitope more available to germinal center B cells. So there's probably enhanced B cell receptor signal, enhanced T cell help with more B cell recruitment into germinal center. And we hypothesized that this nanoparticle adjuvant might be able to overcome immunodominance and stimulate a robust response, antibody response towards the subdominant epitopes. Whether this is true, so we compared alum and nanoparticle, and we immunized mice with influenza hemagglutinin. This is H1 from a PR8 strain, and then we compared. We look at antibody response. First, we coated the plate with this whole protein, full-length influenza HA, and we found that nanoparticle actually is superior in generating antibodies. It's a little better than alum. Alum can also generate high titers of antibodies against hemagglutinin. Then we look at antibodies induced by this vaccination specific to the subdominant stem region or epitope, and we found that most of the mice immunized with alum formulated TR7, they cannot generate antibodies against the stem. And most of the mice immunized with this nanoparticle adjuvant can actually generate very high titers of antibodies against the stem region, suggesting overcoming of immunodominance. And I just showed you that the stem of HA is more conserved between different viral strain variants. So whether overcoming immunodominance can actually increase the breadth of B cell or antibody response against influenza. So first, we did this study. We immunized mice with H1 from PR8, and then we look at autologous response, and nanoparticle is a little better. Then we come to a heterologous. We look at the serum binding capability to a heterologous strain of also H1, but it's a different NC99 strain, and you can see that some of the alum immunized mice start to lose the response, and the antibody titer is pretty low, and all the nanoparticle adjuvant mice can generate high titers of antibodies against a different strain. Then it comes to a different subtype. It's heterologous, but a different subtype, right? H5. So it's not H1 anymore, suggesting it's more different. The immunogenicity probably more different compared to H1. And in this case, most of the mice immunized with alum formulated adjuvant cannot generate an antibody response. Again, most of the nanoparticle adjuvant immunized mice can generate robust response. Then it's a different H3. It's from Hong Kong 14. That is a influenza virus from even a different group, suggesting they are very far away from each other. And in this case, none of the alum group can generate antibodies, but still the nanoparticle group can generate a robust response against this heterologous cross-group variant. Just to confirm, because we just used one H1, we want to confirm the result. So we injected mice with a very different hemagglutinin from a different group, Hong Kong 68, and look at autologous and heterologous response, and we basically got very similar results, suggesting that this nanoparticle not only can overcome immunodominance, probably because it can overcome immunodominance, it can generate a more broad response against many strains of influenza. So how about protection? This is just a binding of antibody to the immunogen. How about protection? So to do this, we immunized the mice with H1 from NC99 with 2 doses of vaccines adjuvanted by either by alum or nanoparticle formulated TR7 agonist. And 4 weeks later, and we challenged the mice with H1N1 PR8, which is a very different strain. And you can see that this is a lethal challenge. It kills most of the mice, unvaccinated mice. And TR7 alum vaccinated mice actually have some protection, about 40%. And it's pretty similar to what we got from the flu shot, right? It's average is around 40% protection. But the TR7 alum, the nanoparticle adjuvanted vaccine almost can fully protect all the mice from the lethal challenge, suggesting much broader protection against heterologous strains. So how about the pathology? So we actually took some of the mice survived this lethal challenge to look at their lung. And you can see this is uninfected mice. And this is the normal lung. And you can see there's the air can actually goes in because this unique structure of lung. And for unvaccinated mice, even though there's one mouse survived this, and their lung looks terrible, so you can tell that it's probably not very easy for the air to exchange in this kind of lung. And for the mice survived the TR7 alum immunization, their lung also looks not very good, and you can see a lot of infiltration of immune cells, and it's hard for the air exchange. And surprisingly, or expectedly actually, TR7 nanoparticle immunized mice, it can protect the lung much better. It's almost similar to uninfected mice. So when we got all those data and COVID came, and we just wondered whether this is also good for COVID vaccine. So to test this, we immunized the mice with the SARS-CoV-2 spike protein from the original Wuhan strain and adjuvanted by either alum or nanoparticle. And you can see that for multiple different variants, the nanoparticle is just much better compared to the alum for inducing the RBD binding antibodies, most of which are neutralizing antibodies. And even for highly escaping Omicron, and most of the mice cannot generate antibody, RBD binding antibody response in the alum group, but the nanoparticle can still maintain a pretty good antibody response, suggesting this is a nanoparticle adjuvant can be used kind of a universal adjuvant, can help deal with the future pandemics. So those are all good. How about longevity of the immune memory, which is very important because we know that we have to get boosted multiple times because the antibody response doesn't last long. And the long-lasting antibody response, antibodies are actually generated by the long-lived plasma cells in the bone marrow. So to test the longevity of the humoral memory, we actually did a mouse experiment last a whole year. So we immunized the mice, and 1 year later, we look at their bone marrow ELISpot assay, and you can see there are few antibody secreting plasma cells in alum group, and there's a lot more in the nanoparticle group, suggesting that it also can promote extend the longevity of humoral memory. So there is a big worry on that. Looks like we kind of alter the immunodominance by providing extra help to the germinal center B cells. But whether the extra help can change affinity maturation, because they compete for to facilitate maturation. Is there too much help? It might compromise affinity maturation, right? So to look at this, we injected mice with NP ova. NP here is not nanoparticle, but it's a hapten attached to ova, and NP here is highly immunodominant because by day 14. You can generate a lot of NP response, but for alum adjuvant, you don't generate any ova-specific antibodies. And again, we see overcoming immunodominance by nanoparticle formulated adjuvant in this. So whether this is associated with change of affinity maturation, so we look at this by testing by ELISA assay, testing antibodies binding to NP2 BSA versus NP14 BSA. So only the high affinity antibodies will bind to NP2 BSA, and most of the antibodies can bind to NP14 BSA. And the ratio of this can actually measure the high affinity antibodies in total, right? So with NP-OVA immunization, usually between week 2 and week 3, and the affinity will drastically increase, between these time points. So we look at week 2 and week 3, and for alum, and it's not surprising that affinity increased significantly between week 2 and week 3. And very interestingly, even though there is a huge germinal center response and there is overcome of immunodominance in the germinal center with a lot more TFH versus TFR, and it really doesn't affect the affinity maturation. It still can affinity mature. So nanoparticle overcome immunodominance, but it doesn't actually compromise affinity maturation, which is very good for a vaccine. Again, a huge problem with many vaccine adjuvant is the adverse effects, and people really cannot tolerate. How about this vaccine, which can generate a huge look like an antibody response? So to test this, we look at the serum systemic inflammation by measuring serum cytokines with Luminex cytokine panel, and this is compared to a naive unimmunized mice. So the blue means there's no change, and red means there's a change. So within 3 hours, you can see the TR7 alum immunized mice. There's some of the cytokines are increased hundreds of times. The mice probably don't feel good, all right? Even though they cannot tell you, but this looks like a mini cytokine storm, even though cytokine storm is in clinical is describing something much more severe, but this looks like there is a mini cytokine storm. It probably will give people a headache or something like that. But if you look at TR7 nanoparticle, probably because it's a slow release, localized release, so it really doesn't change the cytokine that much, suggesting there's no systemic inflammation by this adjuvant, even compared to traditional formulation. So with 24 hours, you can see some of the alum mice still have increased, such as L6, and the TR7 nanoparticle is almost not much difference compared to a naive mouse. So my time is up, but the take-home message is overcoming immunodominance is a promising strategy to broaden B-cell response to pathogens. And the immunodominance can be modified very simply just by changing the formulation of adjuvant. This has great advantage for pandemic preparation because you just need the virus or viral immunogens just mixed with this adjuvant, and it can broaden B-cell response, right? It can probably help protect from future viral variants. And increased breadth of B-cell response, unlike what a lot of people thought, it really doesn't compromise affinity maturation, or it doesn't associated with increased toxicity. Actually, sometimes it can be less toxicity and even better or similar affinity maturation, at least in our case. So now I started my own lab. I really trying my best to understand the mechanisms better, so that help us optimize this adjuvant. So hopefully in future it can be used in some clinical trials. So most of the work are done when I was at Bali Prevention Lab at Stanford University in collaboration with Dr. Qian Ying from Mark Davis lab. Our labs are next to each other, so it's very good to collaborate with this neighbor. And now I started my own lab at Indiana University with help of my postdoc and students. We are hoping look into the mechanisms and optimize the future vaccines. So thank you so much. I really want to thank Sino Biological for this opportunity, and I'm happy to answer any questions. Thank you.
Thank you, Dr. Loa. That's excellent presentation. Very exciting data and technology there. We appreciate you sharing that with us. We've got just a few minutes left, so I'm going to jump right into the questions section of your seminar today. That starts out with a question from Mr. Williams' son. It's a very basic question. I'd kind of like to know the answer to this too, is what makes one epitope more immunogenic than another one?
Yeah, this is complicated. We don't fully understand. It could be multiple reasons. For example, this epitope might be more accessible to B cell receptors, or it's on the, or another possibility is that we might have more germinal B cells that are specific for this epitope than the other epitope. Then it get advantage to be involved in germinal center, and with this small advantage within germinal center, selection, it might quickly gain dominance over other B cells. So we don't fully understand the mechanism, but those are a couple of possibilities. I think probably the reason why some epitopes are more dominant.
Great, thank you for that. And John Kui is asking, would it be possible to formulate a DNA or mRNA version of the vaccine, utilizing your nanoparticle-based platform?
So this nanoparticle is more like can boost the immune response. The current mRNA vaccine are actually formulated by a different lipid nanoparticle, which also have some adjuvant effects. So whether our nanoparticle adjuvant can actually help this lipid nanoparticle formulated mRNA vaccine, that's a question I really want to test. So that's I actually have planned an experiment to look at a COVID-19 mRNA vaccine adjuvanted by our nanoparticle vaccine to see whether it can actually broaden the B cell response to SARS-CoV-2 variants.
Great. Thank you for that, John Rue. Great question there. The next question is from Henri Maynard. Henri is asking, can such a nanoparticle-based vaccine strategy be a problem for a person or an individual that might have an ongoing autoimmune disease like lupus or rheumatoid arthritis?
Yeah, that is a very, very good question. Actually, it's also in our future directions. We are going to test first with animal models which already have existing underlying autoimmunity, such as LPR mice, which is lupus-like autoimmunity. We're going to test those mice to see if this nanoparticle adjuvant can make the autoimmunity worse, or whether it can affect the disease score. If it doesn't, I feel like it probably doesn't, because if you look at the cytokine change, and it really doesn't induce a systemic response at all. Yeah, it's up to test.
Yeah, great. Thanks again for that. And Martin Bukima asks, to what extent are the observed effects derived from TLR7 activation, since the nanoparticles are known to enhance the retention of vaccines on follicular dendritic cells?
Yeah, I think the nanoparticle doesn't directly increase retention of the antigen to follicular dendritic cells. But follicular dendritic cells express high level of Fc receptor. They express high level of Fc receptor gamma 2b on their surface, so they can bind to the antibody antigen immune complex. So I think the nanoparticle can still increase the retention indirectly by promoting a robust early extrafollicular response, which can generate a lot of immune complex, which can be efficiently retained in the FDC network to help the future germinal center response.
Great, thanks for that. And Khalid Dewan is asking, do the nanoparticles induce cellular immunity other than antibody production?
Yes. I encourage you to also read our recently published paper in Nature Materials. This can also leads to increased CD8 T cell response and tissue resident T cell response. That's correct. So yeah, today I'm only presenting B cell part, but we did a lot of T cell, CD8 T cell work.
Great, good to hear that. And David Zawija, forgive me, David, if I mispronounce your last name, asked the question, have you tested the nanoparticle platform without TLR7 embedded in the nanoparticle?
We didn't test those. So that's a good suggestion though. So we don't think the nanoparticle alone can stimulate the immune system, but again, we have to test to prove.
Right. Let's see. So we got time for one more question here. I apologize to the others who won't get to your questions today. This question is from Raja Bilawal Khan. Raja is asking, is there use of different types of IgG and any other types of immunoglobulin interactions with the antigens? I'm not sure if you understand that question.
Not really. Could you repeat?
It says, is there use of different types of IgG and any other types of immunoglobulin interactions with antigens?
Yeah, we haven't measured IgG subtypes rigorously, but what I know is alum can promote mostly in mice the Th2 response is IgG1 dominant Th2 response. With TR7 agonist added, it will switch more to IgG2 to see in black 6 mice. But we don't know whether there is a much difference between nanoparticle and alum formulation.
Great. And a number of people are asking this, so this will just be the last question for you. Can you give us your opinion on like how many boosts or vaccinations would be needed using your system? I mean, despite the fact that no clinical trials have been done yet?
Yeah, we haven't done any clinical trials, but I think with this nanoparticle adjuvant, we probably need less boosts because I just showed you the long-lived plasma cell response. It's really much better just by 2 doses of vaccination. You generate several folds. I can show this picture. Yeah, it's this slide. So the durable antibody response is basically maintained by the long-lived plasma cells in the bone marrow, and this is 1 year after 2 doses of vaccination. And you can see we still have a lot of plasma cells in the bone marrow. So it's many folds compared to the alum. So we don't think we using this. We are hoping to reduce the boosts, which is kind of annoying if you have to get it every 6 months.
Right. Yeah. Great. Appreciate that. We're out of time, folks. I apologize to those that had questions we didn't get to. Literally, it's like 40 remaining questions. So we really appreciate all the good input. We will get your questions answered separately. We have a log of these in your email addresses. So thank you for that. First of all, I want to thank all of the attendees that attended. It's one of our biggest turnouts ever. Dr. Law, thank you for bringing in all these folks. We appreciate it. Just very quickly, we had people, of course, from all over the United States, also attendees from Qatar, Pakistan, Ukraine, Austria, KSA, Iran, India, Canada, Kenya, Puerto Rico, Italy, Yemen, Hungary, Trinidad and Tobago, the Netherlands, St. Kitts, Algeria, and Indonesia. So thanks, everybody. We really appreciate the wonderful turnout today. We know this is kind of inconvenient for some of you given your time zones. Dr. Wall, I want to thank you as a speaker. Fantastic work. We hope to see this nanoparticle delivery and adjuvant system for vaccination in clinical trials soon. Really, really wonderful talk today. And then finally, I want to thank my colleague, Max Bleckman, for setting this up and executing it on behalf of Sino Biological. Max, we appreciate your efforts as well. And with that, I'll just say thanks again, Dr. Lawal, and we'll sign off now.
Thank you, Rob and Max. Bye.
Thanks. Bye-bye.